Sorry about that. I had a phone call, and I didn't want it to end my video, so I had to end my video. Tertiary structure of a protein is just the overall three-dimensional shape that forms when our protein folds, when our secondary structure with all the hydrogen bonds takes on a few more interactions. So we're going to look at these interactions and repulsions that add to the tertiary structure. So the first one are hydrophobic interactions. So whenever R groups that are near each other are of the same polarity, so either being both nonpolar or being both polar, they can have interactions with each other. And so all of our non-polar groups are going to want to kind of clump together and be part of the same environment. And so we're going to get some sticking together of all the non-polar parts. In fact, if you have a protein and you imagine it being kind of in a ball shape, because most of our proteins are either going to be globular, which are kind of, glob-shaped or ball-shaped, or they'll be fibrous. Those tend to be more of the structural ones. The globular ones, if they're moving around in our blood or in our cells, that's an aqueous environment. So you can imagine on the outside, all of the protein parts, right? This is a folded protein. All of the parts, all of the amino acids that find themselves on the outside, they probably have R groups that are polar. All of the amino acids that find themselves clumped on the inside of this ball are all probably non-polar R groups because they are away from the aqueous environment, but also they all cluster together and fold that way because the groups are similar, and so they naturally fold on their own to be in the right shape. So we have hydrophobic interactions. We have hydrophilic interactions. These are the polar groups. The polar groups are going to be on the outside of these globules, because they're going to be wanting to interact with water, with the water environment. We have salt bridges. Salt bridges are going to form between our polar acidic and our polar basic aminos, because those are going to have opposite charges and they're going to see each other, and we're going to form these ionic attractions. So the nonpolar interactions, the polar interactions with water and other polar groups, salt bridges are going to cause these things to link up. You already saw what happens with hydrogen bonding from either the backbone of our amino acid or backbones, groups on the other side groups. The R groups of different amino acids have carboxyls. Different amino acids have OHs. Different amino acids have aminos. That means that they can all form these hydrogen bonding. So we're going to get additional hydrogen bonding between R groups on top of hydrogen bonding between the backbone amino acids. And then we're going to have disulfide bonds. Disulfide bonds form specifically between cysteines. Whenever we have cysteines near each other, remember they can reduce to form this disulfide. And so a tertiary structure altogether is what your protein folds into when we have all of those interactions happening. So if someone asked you what kind of interactions happen that make tertiary structures, you would say hydrophobic interactions, hydrophilic interactions, hydrogen bonds, salt bridges, disulfide bonds. It would be the whole thing. What type of interaction would you expect between the R groups of the following amino acids? Phenylalanine and valine. You'd have to know what kind of amino acids they were. You'd have to say, well, phenylalanine's a nonpolar and valine's a nonpolar, so they're probably going to have hydrophobic interactions, nonpolar interactions. What about between two cysteines, right? That one should be easy. There's only one kind of interaction that happens between two cysteines, or in fact, we could say it another way. There's a certain interaction that can only happen between two cysteines. That's your disulfide bond. Or what if it said, between glutamate and serine, right? Serine has an OH on it. Glutamate has a C double bond O. We can get some hydrogen bond happening here, right? Or if it's two charged amino acids like glutamate and arginine. Arginine has a plus charge. Glutamate has a minus charge. Those would be salt bridge. So you can kind of guess what kind of interaction you're going to have if you just know what are the two amino acids that are important, you know? And that also gives you an idea of why sequence is so important, right? For this interaction right here, this hydrophobic interaction to take place right here, it meant that the amino acid right here needs to be a very certain one. And the other amino acid right here needs to be a very specific one. Because if they aren't the right amino acid, this little interaction isn't going to happen, and we're not going to get the proper folding that we would have. So primary sequence is so important. Quaternary structure is just the activity or the active protein that we get when we have two or more subunits or polypeptide chains that interact together. So a quaternary structure isn't necessary. Some tertiary structures are perfectly capable of doing jobs. In fact, hemoglobin has a cousin called myoglobin, and myoglobin is going to be a tertiary structure only. It doesn't have multiple subgroups. But if you have multiple subgroups or multiple tertiary proteins that all interact with each other, we call that quaternary structure. And so again, hemoglobin has 4 different chains. I should say they have 2 different chains, 2 alpha chains and then 2 beta chains. Now the number of amino acids differs. Compare this, and I always find this to be interesting. Cats, for example, they transport oxygen around their blood. They must have a molecule like hemoglobin. When we actually sequence the gene for their hemoglobin, we see that it codes for a protein that only has something like 40 to 70 amino acids in it. I forget offhand which one it is. Completely different molecule, yet it still does the same job, which is always fascinating to me. So these, sorry, just to come back here. So these subunits are held together by the same stabilizing interactions that occurred in the tertiary. So all the same stuff that held the tertiary protein folded up, those are the same interactions that help quaternary structures hold together. So hydrogen bonding, ionic, salt bridges, cysteine, disulfide bridges, disulfide bonds, nonpolar interactions, all of those. Hemoglobin and myoglobin are actually very similar. They have the same biological function. They both carry oxygen in different parts of the body. Hemoglobin around in our blood, and myoglobin just carries it from our blood into the muscle tissue. They have different molar masses. Myoglobin is only about 17,000 Dalton. Hemoglobin 67,000, so about 4 times bigger. They have the same tertiary structures, and they can carry different amounts of oxygen. Myoglobin can only carry 1 oxygen molecule, and hemoglobin can carry 4. There are advantages to hemoglobin being that way. I don't think we talk about them here. When we get into the enzyme chapter, I'm not even sure if we do in this class. There's something called cooperation, positive cooperation, that happens with the subunits in hemoglobin. When one binds oxygen, it changes its structure slightly, which causes the other molecules to change their structure slightly and be more receptive to binding oxygen. It's kind of help each other out that way. Quaternary structures then consist of the primary, secondary, and tertiary structural levels on top of them being quaternary. Let's talk about some of the side effects of having some misformed proteins, let's say. Sickle cell anemia is an abnormality in the shape of one of the subunits of hemoglobin. And it happens because in the sickle chain, the B chain, an amino acid that was supposed to be polar switches to be a non-polar amino acid. And all it takes is that one change, and that can be caused by one single letter change in your DNA. When this non-polar R group is attracted to the non-polar regions, when valine is there, it gets attracted to non-polar regions of the beta chain, and so this changes the cell shape from something round to something more, sickle-shaped. Or crescent-shaped, and that interferes with the ability to transport oxygen because these sickle shapes end up causing them to stick together. These hemoglobins now with this new shape stick together. They form these other interactions that they wouldn't have before. They form these strands, and then they form these big fibers that end up being very insoluble. Not only are they insoluble and unable to carry oxygen, but they also can clog your capillaries. They can cause inflammation, pain, organ damage. So it's a pretty bad thing. And so luckily, if you have at least some of your myoglobin is working, and at least it depends on how bad you have this. If this is in If you're, what is it, homozygous or heterozygous for this particular disease, you might still have half of your hemoglobins forming okay, while the other half forms sort of the sickle cell. One more thing to talk about. Let's talk about protein hydrolysis and denaturation. So denaturation of a protein occurs, as you can see here, with a few different, well, actually this is showing heat, really. Sorry about that, my kids must be playing with somebody's keys. Interactions that stabilize the secondary, tertiary, and quaternary structures, when we disrupt those, we call that denaturation, and it causes the protein to unfold. When it unfolds, that messes up the shape, it makes the protein no longer active. And there's a few different ways that we can do it, and I'll show you a few different ways. Of course, it says it kind of here. So a denatured protein is not going to have the same function or the same structural integrity that the folded one would have. Now, first, let's talk about hydrolysis. Hydrolysis just means breaking our amide bond. Okay, when we hydrolyze a protein, we get back amino acids, and so you should be able to find the NCC sort of point and be able to hydrolyze these back into their original amino acids, giving them both their ammonium and their carboxylate charges. Hydrolysis happens, in the stomach. It happens through reactions with acid or enzyme catalysts. So, and there's enzymes in our stomach, like peptidase, that would go in there and start to help break these down. Your primary structure is broken up by these enzymes or by just an acid environment, and that covalent bond, that peptide bond, that amide bond breaks. Now, that is hydrolysis, and that needs to be done, before digestion. Denaturation can happen, outside of digestion. So this involves messing up those folded secondary and tertiary structures by either increasing the temperature, making the pH change, adding certain compounds or heavy metals, mechanical agitation. Have you ever beat some eggs? That'll do it right there. Or just manually cause something to no longer look like its shape, right? That's kind of a rough way of like grinding something up. We could do that too. And then there are ramifications, like if it's a globular protein, that means it unfolds, probably doesn't work good anymore. Tertiary structures are disrupted. It wouldn't be biologically active. Sometimes they're no longer soluble. Sometimes they are more soluble. Things just depend on what kind of protein we're dealing with. And so let's look at those examples real quick. So as far as heat goes, when proteins are denatured, because of heat, the hydrogen bonds are what breaks. The energy of the molecules gets so great that it just overcomes that hydrogen bond. It's just like boiling something. Even though hydrogen bonds are there, it's not like it can't boil. This doesn't change the nutritional value. It does make them more digestible. That's why we use high temp to sterilize things, because if there's any organisms on there that we want to get rid of, we can be confident that their proteins are denatured. Changing the pH. Why is this important? Well, this changes hydrogen. This affects hydrogen bonds. If we have a nitrogen that looks like this guy can hydrogen bond, right? He can pull a hydrogen, or he can react with a hydrogen that's attached to an OH, for example, or attached to an O that's attached to something else. This is an OH group. But if this is an acid environment, then he's already going to have an H. And maybe we won't get a hydrogen bond now between these two. There's nothing left on the nitrogen to be looking for anything on the OH. So, and likewise, if the pH changes where we had, an OH reacting or hydrogen bonding to maybe something else, and the pH changes, maybe this H is gone now, and the hydrogen's gone, and so now we don't have that OH again. So it can disrupt those ionic bonds and those salt bridges by changing charges, but also by messing up the ability to hydrogen bond. Tannic acid, a weak acid, is used in burn ointments because it will cause the proteins to coagulate. So it basically forms a scab. And again, that's because it disrupts the normal folding of those proteins. We could use organic compounds. So ethanol, isopropyl alcohol, they basically just go and they hydrogen bond with the let's see, exchanging the bacterial protein's hydrogen bonds to water with their own. So they disrupt the way that those molecules interact with water, just by being sort of like competition for the water molecules. So this just messes up some of their side chains. It messes up some of the hydrogen bonding, and it causes, proteins to unfold. We like to use this on bacteria, right? We like to wipe things with alcohol to kind of mess up the cell walls of any organisms that might be there. And then heavy metal ions. So silver, lead, mercury, these will form bonds with proteins that can't be undone. So that usually messes up with their ionic parts. If this goes and bonds to something negative on a molecule, then that won't be able to go and form a salt bridge anymore. Or it can actually react with disulfide bonds. One of the places where we like this to happen is when babies are born that could have gonorrhea in their eyes. We put silver nitrate in their eyes because that will go and kill the gonorrhea. I think this is a problem when the mother has it. And so this will, destroy that bacteria by basically using that heavy metal, that silver, to go in there and bind to some of the positive or negative, probably the positive parts, the negative parts of that protein, and disrupt the ionic bonding that's holding their proteins together. And lastly, agitation. The whipping of cream and the beating of eggs are examples of this. It stretches the polypeptide chains until their interactions are disrupted. It just basically, we're stronger than a protein, right? So if we move it around enough, we can break those hydrogen bonds, we can disrupt those disulfide bonds, we can break those ionic interactions. All right, so to wrap up, here are the 5 different sort of examples of denaturation we went over. I do have a quick learning check for you. I don't imagine this is going to be very hard. What is similar about using tannic acid to form a scab, or an egg, being hard-boiled by putting it in hot water? And the answer, of course, is that they both are examples of denaturation. The proteins in the egg got denatured, so they're no longer the same that they were. Same thing with a scab on a burn that we talked about. The proteins got denatured, and so now they're all clumped up instead of doing whatever it was that they were doing before. But 2 examples of just breaking those secondary, tertiary, and quaternary bonds. And then a concept map. All right, guys. So we will be talking more about amino acids, I think, and proteins. Proteins at least, in the next chapter or 2. But if you have any questions, you know how to ask them, and I will see you in the next one. All right, guys.